Dear students, welcome to our program. Today, our topic is Concept of Antibody Diversity and Class Switching, Isotype, Allotype and Idiotype, Hybridoma Technology, Monoclonal Antibodies, and finally, basic concepts of Abzymes, Immunotoxin, Chimera and hybrid antibodies. Let's first discuss antibody diversity. The generation of antibodies with different specificities is known as antibody diversity and is created by several antigen-dependent and independent mechanisms. In addition, in B cells that have been stimulated by antigen and received T cell help, Ig genes undergo increased mutational events that may increase the affinity of the antibody produced by the B cell. Overall, diversity is generated by antigen-dependent events at the DNA level as a result of somatic mutation in the V region, which may create higher affinity antibody binding sites. Although rearrangement of the gene segments that will make up the V region genes occurs in an ordered fashion, they are chosen at random in each developing B cell. As these events occur in a vast number of cells, the result is that millions of B cells, each with different antigen specificity, are generated. Additional diversity is created during recombination of V and J of L chain and V, D and J of the H chain gene segments due to the imprecise joining of the different gene segments making up the V region. Furthermore, after antigen stimulation of the B cell, the DNA of its L and H chain V regions become particularly susceptible to the somatic mutation and undergo affinity maturation. Diversity is also generated because of the fact that any L chain can interact with any H chain to create a unique binding site. Antigen-independent events at the DNA level as a result of multiple germline V, D and J heavy and J light chain genes. At the DNA level as a result of random combination of V, D and J segment or V and J segments. At the DNA level as a result of imprecise joining of V, D and J segments. At the protein level because of random selection and pairing of different combinations of L and H chain V regions in different B cells. Dear students, now let's have a brief look at class switching. B cells expressing IgM and IgD on their surface are capable of switching to other H chain classes, for example, IgG, IgA or IgE. This class switching requires stimulation of the B cell by the T helper cells and in particular requires binding of the CD40 ligand on the T cells to CD40 on B cells. In addition, the cytokines produced by the T helper cells influence the constant region gene to which class switching occurs. T helper 2 cells produce interleukin-4 induce B cells to class switch to IgE. Interleukin-5, which is also produced by Th2 cells, induces B cells to class switch to IgA. IFN gamma produced by Th1 cells induce class switching to IgG1. Class switch is guided by repetitive DNA sequences 5' to the C region genes and occurs when these switch regions recombine. The intervening DNA is cut out and the resulting DNA or the rearranged chromosome in the B cell which has class switched and in plasma cells derived from this B cell no longer contains C mu or C delta or other intervening H chain C region genes. Dear students, now let's discuss antigenic determinants on immunoglobulins. The antigenic determinants or epitopes on immunoglobulin molecules fall into three major categories: isotypic, allotypic and idiotypic determinants which are located in characteristic portions of the molecule. Now let's discuss first isotype. Isotypic determinants are constant region determinants that collectively define each heavy chain class and subclass and each light chain type and subtype within a species. Each isotype is encoded by a separate constant region gene, and all members of a species carry the same constant region genes, which may include multiple alleles. Different species inherit different constant region genes and therefore express different isotypes. When an antibody from one species is injected into another species, the isotypic determinants will be recognized as foreign, inducing an antibody response to the isotypic determinants on the foreign antibody. The next is allotype. Although all members of a species inherit the same set of isotype genes, multiple alleles exist for some of the genes. These alleles encode subtle amino acid differences called allotypic determinants that occur in some, but not all, members of species. The sum of the individual allotypic determinants displayed by an antibody determines its allotype. In humans, allotypes have been characterized for all 4 IgG subclasses, for 1 IgA subclass, and for the light chain. The gamma chain allotypes are referred to as GM markers. Antibody to allotypic determinants sometimes is produced by a mother during pregnancy in response to paternal allotypic determinants on the fetal immunoglobulins. Antibodies to allotypic determinants can also arise from a blood transfusion. Now finally, let's discuss idiotype. The unique amino acid sequence of the VH and VL domains of a given antibody can function not only as an antigen binding site but also as a set of antigenic determinants. The idiotypic determinants arise from the sequence of the heavy and light chain variable regions. Each individual antigenic determinant of the variable region is referred to as an idiotype. Because the antibodies produced by individual B cells derived from the same clone have identical variable region sequences, they all have the same idiotype. Anti-idiotype antibody is produced by injecting antibodies that have minimal variation in their isotypes and allotypes so that the idiotypic difference can be recognized. Often a homogeneous antibody such as myeloma protein or monoclonal antibody is used. Injection of such an antibody into a recipient who is genetically identical to the donor will result in the formation of the anti-idiotype antibody to the idiotypic determinants. Dear students, now let's have a look at hybridoma technology. Hybridoma technology is used to produce a hybrid cell. These hybrid cells are produced by fusing B lymphocyte with tumor cell, and they are called as myeloma cells. Thus, these hybrid cells have got the ability to produce antibodies due to the B lymphocyte genetic material and also capacity to divide indefinitely in the culture due to the presence of tumor cell or myeloma cells involved in the production of hybrid cells. Therefore, these hybrid cells produced from hybridoma technology are cultured in laboratory or passaged or subcultured using mouse peritoneal cavity, and these cells produce monoclonal antibodies. And this technology is called as hybridoma technology. Hybridoma technology was first discovered by G. Kohler and C. Milstein during 1975. Dear students, let's have a look at the procedure of hybridoma technology. First, B lymphocytes are extracted from the spleen of an animal, but usually it's extracted from the mouse, which has been immunized with the required antigen against which monoclonal antibodies are produced. Mouse is immunized by giving antigen injection along with an adjuvant via subcutaneously or by peritoneal cavity. This is followed by boosted doses of the antigen. Second, this immunization with specific antigen increases the specific antibody producing B lymphocytes. This considerably increases the chances of obtaining the required hybridoma cells or clones. Third, this specific antibody producing B lymphocytes are then mixed with the selected myeloma cells and are induced to fuse to form hybrid cells. The myeloma cells are selected based on some criteria like these cells themselves should not produce antibodies, and they should contain a genetic markers such as HGPRT. This genetic marker helps in easy selection of the resulting hybrid cells. Fourth, when HGPRT myeloma cells are fused with a specific antibody producing B lymphocytes, the resulting cell population will have the mixture of cell populations such as hybrid cells, myeloma cells, and B lymphocytes. Fifth, this mixture of cell population is then cultured in selective media known as HAT medium along with the drug immunopterin. The HGPRT myeloma cells cannot divide in HAT medium due to the presence of immunopterin. The specific antibody producing B lymphocytes are unable to divide continuously in the culture medium, therefore, eventually they die. Sixth, only the hybridoma cells have got the ability to divide and proliferate on the HAT medium because genome from the B lymphocyte makes them HGPRT positive and genome from which myeloma cells they can divide indefinitely. Thus, only the hybridoma cells or fused cells are selected using selective media called as HAT medium. Identification and isolation of the hybridoma cells. Hybridoma cells producing specific antibodies for the antigen used to immunize the mouse are identified and isolated by the following method. First step, the specific antibodies present in each microville are identified using one of the methods such as precipitation method or agglutination method. Most commonly used and most sensitive and rapid method is ELISA, which is enzyme-linked immunosorbent assay. Step second, wells which contain the antibodies specific to the antigens are identified and hybridoma cells are isolated from these wells and cultured. This ensures that these hybridoma cells have the capacity to produce same single type of antibodies specific to the antigen used. Step third, after these hybridoma cells are multiplied using in vitro or in vivo method. Dear students, now let's discuss the mass production of antibodies. The in vivo production involves injecting hybridoma cells into the peritoneal cavity of the mouse. Then, ascitic fluid is isolated and then antibodies are isolated from it. In vitro method, hybridoma cells are cultured in suitable culture media and then antibodies are isolated and purified. The uses of hybridoma technology. Hybridoma technology is used to produce monoclonal antibodies. Monoclonal antibodies are cells derived by division from a single ancestral cell. Monoclonals are a class of antibodies with identical offspring of a hybridoma and are very specific for a particular location in the body, derived from a single clone and can be grown indefinitely. Monoclonal antibodies recognize and bind to antigens in order to discriminate between specific epitopes, which provides protection against disease organisms. Monoclonal antibodies target various proteins that influence cell activity such as receptors or other proteins present on the surface of normal and cancer cells. The specificity of monoclonal antibodies allows its binding to cancerous cells by coupling a cytotoxic agent such as a strong radioactive which then seeks out to destroy the cancer cells while not harming the healthy ones. Monoclonal antibodies are produced by hybridoma technology. Now let's have a brief look at Abzymes. Abzymes are antibodies that catalyze specific chemical reactions, that is, they function as enzymes. Abzymes are also called as CAT-MAB or catalytic monoclonal antibody. As the name suggests, Abzymes or catalytic antibody are antibody molecules that have conjugated with enzyme to carry out catalysis of substrate. Abzymes are antibodies with variable regions possessing enzymatic activity. Naturally occurring abzymes have been observed in normal individuals, for example, anti-vasoactive intestinal peptide autoantibodies and individuals with autoimmune like DNase Abzymes in systemic lupus erythematosus. In addition, a number of artificial enzymes or designer Abzymes are being developed with any desired enzyme activity and specificity. Abzymes are usually raised in lab animals immunized against synthetic haptens. To date, Abzymes display only weak, modest catalytic activity and have not proved to be of any practical use. They are, however, subjects of considerable academic interest. Unlike regular antibodies, Abzymes degrade the virus permanently. A single Abzyme molecule inactivates thousands of virus particles. Regular antibodies inactivate only one virus particle and their antiviral HIV effect is weaker. Therefore, Abzymes production is one of innovation and their use in HIV treatment may become a future hope for millions of patients suffering from HIV. Dear students, now let's have a brief introduction about immunotoxins. Immunotoxins are chimeric proteins composed of an antibody or antibody fragment derived from the immune system conferring target specificity, that is, fused or conjugated to a toxic protein. These are highly potent and active in very small quantities such that killing a cell is possible even by a single molecule in the cytoplasm. Immunotoxins have evolved with the time and technology and can be generally divided into 3 generations. The first generation immunotoxins were produced by chemically coupling native toxins to antibodies using cross-linking reagents that form disulfide bonds connecting the toxin to the antibody. The first immunotoxins contained full-length toxins, including their targeting domains that are not cancer cell specific. Second generation immunotoxins, like the first generation, were made by chemical coupling methods. As research progressed, toxin autonomic cell binding domains were recognized and removed. The resulting toxin fragment that could no longer bind normal cells was coupled to an antibody. Third generation immunotoxins are made by recombinant DNA techniques and combine variable fragments of an antibody and toxins without their cell binding domains on the same protein. The majority of these immunotoxins targeted antigens selectively expressed on cancer cells. Immunotoxins, together with all antibody-based therapeutics, are a growing field in targeted cancer therapy. Dear students, now let's have a brief discussion about Chimera. Chimeric antibodies contain variable domains sequenced from Hybridoma cell line, which can be combined with any constant domain. New genes have been created that link nucleotide sequences coding non-antibody proteins with sequences that encode antibody variable regions specific for particular antigens. These molecular hybrids or chimeras may be able to deliver powerful toxins to particular antigenic targets such as tumor cells. Finally, by replacement of the immunoglobulin loci of one species with that of another, animals of one species have been endowed with the capacity to respond to immunization by producing antibodies encoded by the donor's genetically transplanted Ig genes. By capturing a significant sample of all of the immunoglobulin heavy and light chain variable region genes via incorporation into the libraries of bacteriophage, it has been possible to achieve significant and useful reconstructions of the entire antibody repertoires of individuals. Dear students, now let's discuss hybrid antibodies. One approach to engineering an antibody is to clone recombinant DNA containing the promoter, leader and variable region sequences from a mouse antibody gene and the constant region exons from a human antibody gene. The antibody encoded by such a recombinant gene is a mouse-human chimera, commonly known as humanized antibody. Its antigenic specificity, which is determined by the variable region, is derived from the mouse DNA. Its isotype, which is determined by the constant region, is derived from the human DNA. Because the constant regions of these chimeric antibodies are encoded by human genes, the antibodies have fewer mouse antigenic determinants and are far less immunogenic when administered to humans than mouse monoclonal antibodies. The ability of the mouse variable regions remaining in these humanized antibodies to provide the appropriate binding site to allow specific recognition of the target antigen has encouraged further exploration of this approach. Another advantage of humanized chimeric antibodies is that they retain the biological effector functions of human antibody and are more likely to trigger human complement activation or Fc receptor binding. One such chimeric human mouse antibody has been used to treat patients with B-cell varieties of non-Hodgkin's lymphoma. Well, dear students, that's all in this topic for today. I hope you have understood it. Thank you.